Hello, everyone, and welcome to today's session. My name is Marta Dejia. I'm a SYNGAP parent and director of the Syngap Research Fund. We are very excited to continue the SRF Syngap Research Fund webinar series. The goal of the series is to empower your communications with clinicians as you get more clear knowledge of Syngap. We also want to give you a plug to our next presentation, types of seizures and EEG patterns in SYNGAP1, which will take place on May 27 at 11 a.m. Eastern Time with Dr. Angel Aledo Serrano. Our talk today is functional assessment of missense variants of SYNGAP1. I have the pleasure to introduce today's speaker, Professor Kurt Haas. He is a professor of cellular and physiological science at the Faculty of Medicine at the University of British Columbia. The primary goal of Dr. Haas' research is to understand how brain circuits form during early development and how mutations and errors give rise to dysfunctional circuits that underlie common neurodevelopmental disorders such as autism spectrum disorders, and epilepsy. The distinctive feature of his research approach is the development of image tools that combine electrophysiology, genetics, and microscope designed to see brain neuronal network activity and growth. A recorded version of this webinar will be available on the SRF website under webinars on the family menu. By the end of the presentation, you will have the opportunity to get the answer to your questions. We'd love to hear from you. Please write your questions in the Q&A. For those of you just joined us, welcome. And again, our speaker is Professor Kurt Haas, and his talk is Functional Assessment of Missense Variants of SYNGAP1. It is now my pleasure to turn things over to Professor Haas. Welcome, Professor.
Thanks, Marta. And thanks for this invitation to speak today. The first thing I'm going to do is change my title a little bit. And this is the new title, which is an email I received from Hans Schlecht, which is, I think really puts the work in a better context, which is, are SYNGAP missense mutations simply haploinsufficient? And I'd like to, I think throughout the talk, I'll be sort of really just coming back to this question. So my interest is to understand the genetic architecture of autism spectrum disorder and other neurodevelopmental disorders. And the search for genes associated with these disorders was conducted, has a long history, and the early sort of low-hanging fruit were discoveries of large either gene duplications or large mutations that clearly knocked out protein function. However, things have become much more complicated since it's been realized that the most common mutations are de novo single nucleotide mutations, and that they occur in hundreds of genes that are associated with ASD. So there are really thousands of these mutations. Now, these mutations, they cause single nucleotide changes in the base code of DNA, which can give rise to three different types of mutations. Nonsense mutations, where there's a stop codon inserted so the protein is truncated. Frame shifts, where you have a microdeletion or addition, so all the codon is frame shifted downstream of that point, so the protein is sort of a nonsense protein. Or the most common are missense mutations, where a single nucleotide change causes a single change in the amino acid within the protein. Those small changes are very hard to understand how they impact protein function. So most of them are termed VUSs, or variants of unknown significance. But my lab is interested in trying to really understand what they do. So our strategy, which I think is somewhat unique to our lab, is to develop large platforms that use multiple assays to study individual proteins for what we call deep phenotypic profiling, to understand what individual mutations do to very complex aspects of protein function. Proteins are complex little machines. They often do multiple different functions. And if there's a mutation in their protein structure, it's not obvious or clear that what function they might be interfering with. So we think you really need multiple assays in order to really understand what's going on. Our goals are multifold. One is to discriminate variants that are not likely pathogenic from those that are. Just because we find a variant in an individual with autism or other disorders doesn't mean that it's causal. So by conducting multiple assays and finding that a variant doesn't really disrupt protein function, we think that adds evidence that it's likely benign and non-causal, but other ones likely are. And for ones that are, it's very important to understand what the molecular mechanism of dysfunction is. To do that at the protein level and then the protein sort of pathway level. And then once we identify those, we can take a subset of those to move forward to lower throughput assays, such as for testing for pathophysiology. This is understanding what's happening at the neuronal and neural circuit and whole body level. And eventually this will allow us to develop therapeutics. Having a wealth of knowledge about point mutations throughout a protein also provides invaluable structure function analysis or information about what proteins really do. So ultimately our goal is to link gene mutations to molecular dysfunction and pathophysiology, and all these to disease expression and therapeutic outcomes. And together, this is really the foundation for personalized medicine. And this sort of approach is sort of diagrammed here. And here you have individuals with autism spectrum disorder or other neurodevelopmental disorders, and they usually present in a spectrum with very complex and a range of different phenotypes. And if you're interested in understanding what the cause might be, the strategy is to, then sequence these, the genomes, find mutations. And at this point, largely we're stuck with relying on predictive algorithms to determine whether these variants are deleterious or not. And while there's been really impressive power in these algorithms, they're not perfect. And they should not be a substitute for real experimentation. So this is where we come in and test these variants at the protein level and the protein sort of circuit level. And data can then be fed back to help develop or improve predictive algorithms. And once we've used these methods for high throughput analyses of many variants, we can find sort of representative variants that are representative classes of dysfunction, which can then be brought forward to these sort of slower throughput assays for understanding pathophysiology, what's happening at the cellular or animal level. And then all of these sort of this information at these different levels are useful for developing targeted therapeutics that are specific to specific molecular dysfunctions, specific pathophysiologies, and then feedback to individuals. So this is where I'll be talking about today, this sort of level of just dealing with variants, protein level, and sort of the protein sort of a molecular pathway level. And we're trying to understand what are the classes of the protein dysfunction. And we talked about haploinsufficiency. Typically that means the protein is just absent and it's just sort of gone. But we often talk about loss of function, and this could be partial loss of function or complete loss of function. But to be clear, that function is under whatever you're calling as the function. So there are many functions of proteins. There's also gain of function and dominant negativity. When we look at what proteins actually do and look for dysfunction, this is a short, maybe not even complete list of all the possibilities of what might be happening. There can be impacts of missense mutations on protein stability, subcellular localization, post-translational modifications, so phosphorylation, myristoylation, pulmonylation. There's a tremendous amount of these modifications that regulate protein function, and they can be disrupted. Many proteins dimerize, they bind to themselves, so that could be disrupted. But certainly most proteins bind to other proteins. So there's a lot of protein-protein interactions. If they're enzymes, then there's both catalytic activity and substrate interactions. So any of these processes can be impacted. It can be one or a combination of them. So these are the methods that we use in my lab for high throughput assessment of variant function. We construct large DNA libraries of individual genes, between tens to hundreds of point mutations. We physically make them, and then we put them into expression vectors, which are plasmids we can put into cells and they'll be expressed. And we put them into these sort of genetic tools. And this one is an important one we've developed, which drives two proteins off of the same promoter. So it uses this P2A internal, it's a self-cleaving peptide from viruses. What allows us, this is the promoter that drives first expression of the first gene, which is a red fluorescent protein, RFP. It reads through the P2A, and then that self-cleaves, but then makes a copy of the downstream protein, which is a green tagged protein variant, whatever our gene of interest is. Here's P10. So that would come out as a fusion with GFP. And this is very powerful because it allows the red to be used as a protein expression level, and then the GFP tag to show us where the protein is or how much the protein, if it's degraded. Then we express these vectors into human cells, such as human embryonic kidney cells, HEK cells. These are really workhorses, cell lines in labs that can be grown whenever you want to, easily transfected, and then used for experimentation for expression. And this is the readout. We use fluorescence imaging, and that's either with high content imaging or flow cytometry, and we're detecting either the fluorescence of these red and green fluorophores or applying antibodies that detect typically phosphorylation states of downstream protein markers. So proteins are typically in molecular pathways where they can then activate or suppress, inhibit downstream proteins. And that could be a marker of the protein's function. And by having antibodies to the activation state of those downstream proteins or markers, we can detect protein function. So we'll have fluorescently tagged antibodies, and we'll look at them in individual cells. So on the high content imaging, we are imaging these fluorescent HEK cells. And this is done in an automated fashion, very high throughput. And then there's computer tools to segment these cells and detect fluorescence within them and make large measurements. So you can really move through large numbers of cells at once. And then the other tool that we use a lot is called flow cytometry. Here, these cells are being placed into a solution, and then they're passed one at a time through a detector. Lasers are shot at them and the fluorescence is detected. So we have single cell resolution of all the markers that we're interested in. I'm going to start by not talking about SYNGAP1, but talking about a different protein, P10, that I think gives us a good idea of what our approach is and what one can learn. And then I'll move to our SYNGAP1 studies. So for P10, we recently published a paper last year where we studied 127 variants in 18 assays in 5 model systems. So this is really deep phenotypic profiling. And now we're actually up to 21 assays in something on the order of 150 variants. P10 is a really well-characterized cancer and autism associated gene, and it acts to pretty much inhibit this protein here, AKT. AKT promotes cell growth and proliferation. So by inhibiting it, that's how P10 inhibits cancer, and mutations in P10 causes dysfunction or overgrowth in neurons, leading to autism issues. So what we've done is, down here is the P10 gene with the different domains, and all these lollipop figures are missense mutations that we've made individually on different copies of the gene. And they're color-coded, where the blues are found in individuals with autism spectrum disorder or intellectual disability. So that's this group, the blue group, and you can see they're found throughout the protein. And the other important groups would be these biochemical controls. These are ones that we know interfere with function. And we also include these purple ones, which are variants found in the normal population. So, and this is what the data looks like. This is a flow cytometry sort of data set. Each point here is the response from a single cell. You see there's tens of thousands of cells. This is actually results from 3 experiments laid on top of each other from these 3 different variants of P10, wild type, a hyperstable one, and a unstable version. And what we're plotting is the red fluorescence and the green fluorescence for each cell. Again, the red fluorescence says how much of the protein, the variant protein was made. The green reflects how much still remains as the protein gets turned over. So taking the green to red ratio tells us what the stability of that protein is. And that's shown here in the right. The sort of medium purple is the wild type. This is the plotting the GFP, the green over the red, and this is sort of frequency from these sort of plots. And the salmon color is a destabilized variant that's shifted to the left. The dark purple is a hyperstabilized one shifted to the right. So that's the data set. And this is the data from 127 variants. So this is protein stability. This data is all normalized to wild type. So this is the wild type P10. And I'll just have you focus on first this group here, which are the blues. These are the variants from autism or intellectual disability. And the results were pretty striking. This is the first gene we really looked at. And I was sort of expecting just complete instability or wild type stability. And that's not what we saw. We found variants that were wild type levels. Some were hyperstabilized. And then there was this full range of instability. Again, all these are induced by single point missense mutations. So that was really dramatic. And this really gave, sort of support to maybe this haploinsufficiency model that the disorder is really due to gradual, depending on the case, a gradual decrement in how much P10 is present. And that sort of fits. Interestingly, if we look at these purple ones over here, these are the population variants. Many of them are wild type, but some are a little bit hyperstabilized and a couple are destabilized, showing that even in what's considered the normal population for these databases, there is protein variance in protein function. Okay, the next thing we did is to look at function by looking at the activity of this downstream protein, AKT, using antibodies against the phosphorylated state of phospho-AKT. And that's showing the flow responses. Again, the medium purple is wild type. In this case, the C124S is a known non-functional P10. So that's shifted to the right. So there's more phospho-AKT, and a gain of function for A shifts it the other direction. And this is the data from large numbers of variants. And again, if you look at these blue ones, the autism-associated variants, this was really striking. This shows you the function of these variants. And again, it's a very complex profile. It's not all or none. There's a gain of function that's similar to this one here, which is this known gain of function is for A. Here again, all this data is normalized to wild type is 1, and the empty vector GFP by itself is 0. So, so we have a gain of function. We have a few that are wild type like, and then there's this range of partial loss of function, complete loss of function, and then these, which are dominant negatives. So they're worse than having an empty vector. So this is really important. Only 5 dominant negatives were known before this. We found 27 additional ones. So this suggests that there's a large complexity in the molecular mechanisms of dysfunction for this gene. And what was important was when we went back and compared the protein stability data to the functional data, that's plotted here, where stability is in the x-axis, protein function as a readout for the downstream levels of its ability to inhibit phospho-AKT. You see that we found that there's 2 groups of variants. Each point here is a different variant. There are those, the blue ones, where the function, the amount of function or dysfunction tracks directly with how much stability or instability there is within the protein. So for these, I think that the molecular mechanism is haploinsufficiency. So it's just an absence of the P10. However, the red ones, the amount of dysfunction is not correlated to the amount of stability. So a different mechanism is going on. And if we look at these red or green variant populations along the length of the protein, we see that the red ones fall into specific domains. And these are known substrate binding and catalytic domains. So it really says that, yeah, if there's a variant within these domains, it's not going to be haploinsufficiency that's causing dysfunction. It's because of a disruption of enzymatic function. Okay, so that's, I think, an important sort of perspective in general. We next move to studying SYNGAP1. Clearly, there's many single nucleotide variants of SYNGAP1 that are being discovered. These include nonsense, missense, and friendship mutations, and it's associated, as you well know, with ADHD, epilepsy, and schizophrenia. But even recent reviews, such as this one, this is a direct quote from this paper in 2019, talks about de novo variants of SYNGAP resulting in haploinsufficiency lead to a defined phenotype, and that this may explain up to 1% of ID cases. And I just, I'm a little more cautious about making this sort of statement. And I wonder whether other mechanisms besides haploinsufficiency might be happening. So this is a diagram from a review by Clement, just showing you some of the molecular pathways associated with SYNGAP1. SYNGAP1, the gap is it's a synaptic gap. It's a GTPase regulating protein, and it's known to regulate both RAS and RAP. These GTPases, these pathways, molecular pathways, are very complex, and when you start putting GTPases in them, things get very, very complex. But so this is really a subset of a very small subset of probably what's going on. But you can see there are downstream proteins such as ERK and P38 MAP kinase. And then downstream of both of those is CREB. So, and I won't go through this sort of function, like what SYNGAP1 is doing, because you probably are well aware of this, but it's associated with both neuronal growth and with synaptic plasticity. Very well connected to LTP, lots of work from Rick Hugenier's lab, but it's also connected as well to LTD. So it's sort of in this balance between the two, where the RAS pathway is more connected to LTP and maybe the RAP is more connected to LTD. So we wanted to look at markers downstream of these pathways. So we picked the reds are where we have antibodies against phosphorylation state, detecting the activation of these downstream proteins. So we have markers for activated ERK and P38 MAP kinase, as well as a much more downstream transcription factor, CREB. We also did a quick screen for additional proteins that we know are downstream of RAS, and we found that GSK3 beta is also regulated by SYNGAP1. So we've included that in our screen as well. Just to quickly review the structure of SYNGAP, it has multiple domains, including these plextrin homology or pH domains that are likely involved with membrane recruitment. There's C2 in the GAP domain, which is important for the GAP GTPase activation activity. And then there's this large C-terminal domain that's called a structurally disoriented or disordered domain. Less is known about this. Certainly it's known that there's these SH3 and a coiled domains that are involved with protein-protein interactions. So that's the structure. And these are the variants that we've identified. We found 57 variants of SYNGAP1 that we've included in our study, and we've developed 7 assays to test their structure function. These include a wild type version, 23 that are in individuals with ASD or ID. I call these unconfirmed because, again, just because they're identified in these individuals do not mean that they're causal. Even though we're calling them this, we don't think that they necessarily are causal. We also included 16 population variants. These are from the NOMAD database. So this are thought to be non-disease associated, sort of normal population controls. And those are the blues are the ASD-ID variants. You can see they're found across the protein. And we picked the green ones, the population controls, as being across the protein as well. And then we included 17 what we call biochemical controls. These we think are likely to be loss of function or gain of function. Either, it's known, like most proteins, that SYNGAP1 is phosphorylated, is probably more than 20 sites. And these are the kinases that phosphorylate them. So we picked sites throughout SYNGAP that are the sites for phosphorylation, and we mutated them, as well as loss of function variants that were studied in non-human orthologs. So we include as many as we could. Some we just developed on our own, hoping that they might be non-functional. And this is the data. So this data is presented differently than the data I showed you with the P10. Here, we're not normalizing the data. So I'm always going to show that this is just pure stability. The black is the wild type stability. And so it's black. Red is GFP. That's not very meaningful in stability sense here. So don't, but it is more informative in other plots. And you can see the spread of the 57 variants that we tested. Again, the blues are the autism ID associated variants. The greens are the population controls, and the purples are the biochemical controls. And you can see that there's that in general, SYNGAP1 is resistant, or largely resistant, to missense-induced destabilization. And this is likely because it is such a large protein. It's like 1,300, 43 amino acids. That there are studies that show that longer proteins are less sensitive to instability, though the mechanism for that isn't completely clear. But that seems to be true here. So there are only 3 missense variants here that show significant instability. These two are nonsense early stops at 687, 579. They show strong instability, but all nonsense mutations should not be considered to be destabilized. There's another one right here that is indistinguishable. It's not significantly different from wild type. It actually shows about 88% of wild type stability. So this is likely present. So I wouldn't say that nonsense mutations have to be fall into this haploinsufficient category. They're still present and they might still have partial function. And then there are these 3 over here that are hyperstable, 2 autism and 1 biochemical control. So hyperstability is another mechanism. And when we look at where the very rare ones that are either hyperstable or unstable, they fall within these sort of discrete domains, either in the gap domain or the C-tail region. Next, we looked at function. So this was looking at antibodies against phospho-ERK. And again, this data is not normalized. The black is the wild type. The red is the empty vector, just GFP. So this would be loss of function. All the black stars represent significant difference from the wild type. The red ones are significant difference from the GFP. So what we find is that wild type SYNGAP reduces phosphorylation of ERK by 28%, and there's complete loss of function in 18 of the 57, partial in 22 of them. So, and the lowest ones here are sort of biochemical controls, and as well as the highest ones. So the highest ones are more functional than, or they're equally functional than wild type. Okay, so if we plot out sort of ERK function across the protein structure, it's very clear that there's a distribution. So what's shown here is the GFP value, that's the line here, and the wild type is this black line. So variants that are closer to the black line are more functional. Those are closer to the GFP are less functional. And you can see that ones near the N terminus, none less than amino acid 485 show less than 50% function, whereas those that are above 800, all of them show significant dysfunction. The next assay we look at is GSK3 beta. These results correlated well with the data from ERK. You can see this, and so there's a significant correlation between these 2 markers. So this is important because it shows a novel interaction with a new sort of downstream pathway or RAS pathway of this important protein. GSK3 beta is important both in synaptic plasticity and neuronal growth as well. Then there's the other pathway, that the sort of RAP LTD pathway for the marker P38 MAP kinase, we find less but certainly significant impact. We find the results are correlated somewhat with the ERK results, but there's a lot of variants that are either show more function in one pathway versus the other. And this raises the possibility that different variants are selectively inactivating an LTP pathway versus an LTD pathway. And so it's important to understand what that might result in at the pathophysiological level. And then finally, CREB, which is this downstream transcription factor. We found that while there was some dysfunction, we find that that all variants were able to inhibit CREB phosphorylation compared to wild type, compared to GFP. So you see that they all show some activity, but some show partial loss of function, and then there is some gain of function phenotypes over here. So it's interesting that even in cases where ERK variants don't suppress ERK phosphorylation, they all can affect this downstream CREB, suggesting that maybe it's working through different multiple pathways. I won't talk much about this next data set, but we've also looked for subcellular localization of SYNGAP1. Of course, SYNGAP1 is very important because it localizes to synapses. And one can make the argument that, well, what's the utility for studying this in a non-neuronal system? We think the pathways are still intact, so that's important. But when we looked for subcellular localization, we also found a phenotype. So on the top these are hex cells just expressing GFP, and you can see the GFP throughout the cytoplasm. But when we express wild type SYNGAP, we find that it localizes to puncta in the cytoplasm. And when we look at different variants, we find that there's either a decrease or an increase in the size of these puncta. So it raises, well, a question of whether this is important for its function in neurons. This is the mean speckle size, or the puncta size. This is the wild type. And you see that most are similar, though they are significantly larger than wild type. Some are significantly smaller. And then there's a subgroup that are all significantly much larger than wild type. And these fall into the two early stop variants we studied, as well as two variants that are very unstable, and three additional ones that are very close to the C2 domain. So something very interesting is happening here with these variants. And this shows where they're located across the protein. So when we have this large data set with 7 different assays, we can then do clustering analysis to see which variants are similar to each other, to try to see if there's uniform molecular mechanisms. And this shows an example where here we have one, this R596A, which is a biochemical control, is very different from every other variant. So it's important to find those. And then we can find ones that are very similar. So these two here, which are R485A and M759V, and this other group here, these are the most similar to each other throughout the entire group. And what's important is that even though they're so similar in their phenotypes, they're very far apart from each other in the protein. So, of course, proteins are three-dimensional, have three-dimensional tertiary structures. So it's important to identify these to try to understand why they're giving such similar molecular phenotypes. From this type of clustering, we can identify variants. We can reduce this data set to a smaller number of representative variants that we can then take forward to lower throughput assays. And this is also aided by PCA, or principal component analysis, using multi-dimensional clustering, using all of our data sets. And using this approach, we find 5 groups of variants, and it's driven by these different features, whether they're likely loss of function across assays, likely wild type, or other features. So this is a very important approach for reduction of the variant class. And I'm going to just sort of wrap up by showing this, which is a heat map of the response of each variant for all of our assays, our 7 assays, including stability as well as a multifunctional score, which is an unbiased, just sort of a score that takes into each of these assays with equal weighting. So I think one could debate the utility of this, but I think it's a first pass, it's what we're using. But when you look at these patterns where white is wild type function and blue, or specifically dark blue, is loss of function, what's striking is that there's a mosaic or speckle pattern across these variants, where it's not just all blue, it's not all or nothing throughout different assays. And that's highlighted by these dots that show dysfunction in these markers. And it's very important where you take one like here, where you have strong impact or dark blue in ERK and GSK3 beta, but you have function in the P38 assay, or this one, or this one, or this one. The ERK and GSK3 beta are fine, but there's dysfunction in the P38 pathway. And this one is selectively sensitive to CREB. So this really highlights or suggests that there's multiple mechanisms of dysfunction that have differential impacts in their molecular circuitry. So when we talk about haploinsufficiency, it's hard to sort of reconcile that. And is it possible to say, well, is there function-specific haploinsufficiency that makes sense? So I think a deeper, more sophisticated approach for describing these variants is probably important. When we look at the association of variants to their different sort of groups, again, these is ASD or ID, our biochemical groups, or our population controls, and this is the multifunctional impact score. And it's probably important to do this sort of analysis with each of the different assays as well, but we're just showing you this data. If you see this, it's very clear that the dysfunctional ones down here we think are likely causal, but there are other ones that are very wild type like, and we think it's possible that these might not be causal. They just have to be found in individuals with ASD or ID. Our biochemical controls, some of them are very impactful. It's very important to identify these to allow them to be brought forward as very solid controls for studies of pathophysiology. So it's important to identify the ones that are really loss of function. And then it's important to also realize that in the supposedly normal population, there's a spread of protein function. So it's not, everything's not just one like wild type level. So it's possible that these people have not yet been identified as having disease, or they might just have a difference of susceptibility to disease. And what's shown here are the categorization of these variants found either in databases like ClinVAR or the literature. So pre-assigned associations as pathogenic or likely pathogenic, benign, likely benign, versus variants of unknown significance, or those that are not scored. And you can see that using our multi-functional scoring, we find that there's justification for some, but other ones we think should be looked at, because these ASD ones we don't think are likely pathogenic, whereas the ones declared as being benign, we think there's a lot of dysfunction here, so they should be revisited. But we can also aid in providing some maybe classification of these VUSs or those that are not yet scored. So I think with that, I'll end with this sort of conclusion. Going back to this question, are SYNGAP missense mutations simply haploinsufficient? Well, we find a diversity of impact on downstream markers in both ASD-ID and the normal populations that suggests that there are multiple molecular mechanisms of dysfunction. And importantly, this might differentially impact these important synaptic pathways of LTP versus LTD. We would expect that this might give rise to different pathophysiology. And importantly, we find minimal impact on protein stability. And I think this is impact on protein stability would give the most justification for using haploinsufficiency as a mechanism. So for moving forward, we've created this platform, and it's really, we just see this as the start. We can always add new variants and add more assays. So we're always searching for new variants to test. And I think we have to expand our assays to either known or unknown downstream markers. We need to then take forward our most impactful variants, our representative variants of different classes, to studies of pathophysiology in neural cultures and animal models. And we really need well-detailed clinical assessments to link these specific molecular mechanisms to specific patient phenotypes. So that's it. I'll just thank my lab and my generous funding from the Simons Foundation and from the Canadian Institute of Health. All right, thank you.
That was great. Thank you so much. That really was very good. And yeah, and we have the first question is how you close the saying that you guys need more variants, and we get that question very frequently about getting new variants and getting more kids tested, and how are you guys doing that process?
You know, we research the literature. We have close contacts with the Simons Foundation, other foundations, but we're always open to reach out to groups like yours to find these variants.
Okay, but there is a process that you guys follow through, like blood samples, or because we are going to have
no, so we don't do any of the sequencing ourselves. We always get our variants from other sources. And ideally, there's, you know, good clinical assessment that goes along with them.
Okay, yeah, we have some collaborations very similar. Yeah, that's one question we get along. And I think Hans is here, and Jenny, JR is here too, and they probably have more important questions that I have for sure. Hey, Hans.
Hi, can you hear me okay?
Yes.
Dr. Haas, that was fantastic. I mean, you know, the paper itself is as dense as cheesecake to get through scientifically. So you know, thank you for the missense parents and your team, because it takes a stunning amount of time by everyone lined up here for the photo. I put some questions in the chat and sort of flipping back for screens. You know, the first one, I guess it's remarkable that a couple of variants had significantly increased protein stability. Now, granted, that comes with a level of dysfunction physiologically, but could the two be separated? You know, I'm thinking about my son who has a nonsense variant, where potentially promoting protein stability could, you know, correct haploinsufficiency by a certain percentages, hopefully to benefit the phenotype. So I guess that's my first question.
Yeah, that's a really important point. And, you know, I think every protein is unique in its sort of functional range, the range of protein concentration that's necessary. And it's also probably unique in whether if you have too little or too much, if that matters. And yeah, I think that's something that we have to determine. You know, if we find variants that are hyperstable, I guess we should really look closer to see if those are the ones that are having gain of function in our assays, and test those with pathophysiology. I think that's what has to be done. And I think if you have treatments that increase stability, I think, you know, you definitely want to do a pathophysiology study to see how much are you increasing it, and how that impacts function or measures.
Just in the past 6 months, there's been, you know, headlines about AlphaFold in terms of predicting tertiary structure. And you brought up tertiary structure. Is there plans to try to correlate, you know, that genotype, phenotype to tertiary structure?
Yeah, I mean, that's a really important and work, and it's an area that we are moving into, but we're just developing our expertise. And we have a lot on our plate right now. So, but I think probably just find the right collaborators to work with to hand our data to. So I think having this type of deep profiling is very valuable for modeling, for both bioinformatic, you know, predictive assessments, but that type of tertiary structure. Yeah. So we have to reach out and make those connections.
Let me say thank you again and let JR ask her questions.
Hi, thank you. Yeah, thank you so much, Professor Haas. That was amazing. I'm sorry, I don't quite understand the stability aspect. Did you is it a steady state expression that you're looking at, or did you pulse and look at a time after? So is
No,
that,
it's steady state. I mean, that's a good point
so,
too.
so would you call that persistence?
So,
Or
yeah,
no, you're
abundance.
calling it stability?
I mean, abundance would probably be a best.
Okay, yeah, okay, abundance, that makes sense. So have you, I don't think you did this, but could you ever like heat the HEK cells a little and actually look at the, like stability to me is more like heat stability, that kind of thing. So I wonder if you could just up the temperature a few degrees.
That's an interesting idea that we've considered. That there's complications with, I mean, other things that happen, and there's only certain ranges one can do with HEK cells, but you can definitely change their temperatures. And yeah, we probably should get around to doing that.
It's great that you're making these high throughput assays that can really, look at a lot of things. So thank you for that. And I think we're all going to be really super interested to see so you showed us some graphs about, what was it? What did you call them? At the end, and you said you only had the main one, but you wanted to do it for each assay, like that kind of thing.
Score.
Yeah, the multifunctional score. But looking at that for each assay, I think will be really interesting and making making a picture of what seems to be happening at different points along the protein.
Right, exactly. Ideally, we'd have very nice clinical assessments, and we'd be able to link up specific molecular pathway dysfunction to specific phenotypes. That'd be the most ideal situation.
Yeah.
I think that's what the field needs, and hopefully we're all moving towards.
Yeah, so when we need it all linked up to patient data, right, to see what's going on. Yeah, I guess I'm just really surprised at how much the genome AD, the population variants were right in there with everything else. And so that made me wonder if really just focusing on the ends, the ends after the, so here's, if green's sprinkled in there, just like focusing on the part where there isn't green, if that is going to be more predictive.
Yeah, I don't know. My feeling, I feel a few different ways about this. One, especially in autism, like SYNGAP1 is definitely one of these genes where if you have a strong loss of function, you're very likely to have a problem. The other genes probably less so. But certainly in autism, we think that there's a lot of polygenic combinatorial effects that go on, and that all of us likely carry mutations that predispose us for this, but it's really the combinations. And I think I don't think that they're insignificant. I think that they're significant in the right context.
Right. Well, and I know that when I was first hearing about SYNGAP, I was hearing that less severe, so missense mutations can be associated with bipolar disorder, schizophrenia, and autism without intellectual disability. So I'm wondering at what level, and you even mentioned that, you said, well, maybe people have an issue that has yet to be diagnosed, and some of those things are diagnosed later in life. So
yeah, that's a very important point. Yeah.
I don't think you can do this high throughput, but I'm going to ask anyway. So SYNGAP is a developmental gene, and so it's going to affect the structure of the brain from the get-go. And so to my mind, some of the variability could be in brain structure, in how well parts of the brain are connected to each other, cell morphology, and that's all, maybe pre-birth, right? And then after a mouse or a human is born, then the SYNGAP variant is going to make the brain work differently, right? Like our brains are experiential. We build our brain by what we experience, and so it's going to be built, slightly different that way too. So I'm just wondering, are there any of those? Is there anything there that you think you could look at? Is there cell morphology, or that's just not going to be high throughput?
Yeah, so that's what I call pathophysiology, right? What's happening at the real neuronal level, both synaptic structure and then circuit and in intact organisms. And we do some of that work in my lab, and we have collaborators that do this in different model organisms, and it's not high throughput. It's very laborious.
Yeah,
and this is where what I think is important is that from our high throughput work, if we can identify if we study 60 or 200 variants, but we can reduce that to 5 or 10 representative variants that are very common to the other ones, and move those through our lower throughput assays, may be informative, transferable to the other variants that are within that class. So that's our strategy for trying forward on this in a way that's useful.
Yeah, well, I think we would agree that we'll miss less. We'll miss less important things if you look at all these assays. So, because they've already been somewhat surprising.
Yeah.
So, what would you say for us moving forward when we have people who have a missense mutation and we're not sure what it is? Like, do you have any sort of practical advice for looking at of us?
Obviously there's predictive algorithms, but you have to know which of those really fit SYNGAP. So some clearly don't for SYNGAP. It's very strange. So I think what we're trying to do is use our data and really find the predictive algorithms that are best. Eventually in long term is try to improve that, those algorithms based on our studies. Yeah, I don't know. I think, that's as far as therapeutic approaches, I think one just has to keep an open mind to different molecular mechanisms of dysfunction.
Yeah, but at this point, I have a feeling you're not necessarily, or maybe you are open to us like emailing you and say, what do you think of this one? I mean, what do we, is that something we can do? Or is that something that is coming later and you're like, yes, in 2 years, I'm going to have this algorithm and it's freely available to geneticists, genetic counselors, or?
I think really practically, we're very open to receiving new variants. Ideally, they'd be put into a database like ClinVAR so we can reference them, saying this is a referenced, this is the site, and it has some clinical assessment associated with it. So that's the best thing we can handle. And ideally, we'd repeat this study with more variants and with more assays. So like as I said with the P10 project, we're now engaging, that's developing. Even though we did 127 variants and 18 assays, we've developed more assays, and we're going to put in probably another 100 variants maybe. So these things this platform is established and now can develop as long as we have the people to do it. It is labor intensive, but that's what I see is being able to sort of perpetuate this. At the same time, working with, pathophysiology studies and helping with predictive algorithms and all this. So.
Okay, thank you so much. It's a lot of interesting work.
One of the things that catch my attention was that you showed some variants that they were classified pathologic, but then when you did the function analysis, you said they are not likely to be pathologic. They are not likely to be causing SYNGAP mutate. I mean the phenotype. Then how often that happen?
It does happen. It's happening with our P10 studies. So yeah, this happens. So how variants get classified as pathogenic or benign sometimes it is not very well done, I guess. Other times it's better. And the sort of the boards that make some of these decisions, they don't rely on functional assessments like laboratory assessments typically. There's a standard that a functional assessment is only one component of deciding whether something is it falls into those categories. And that requires, under their guidelines, they'll say, well, a functional assessment for like P10 would only be associated with the phospho-AKT pathway and nothing else. No other functional assessment matters to them. And so it's a very simple approach. I don't think takes into the complexity of protein function and the relation of those functions to disease. So this is another area this classification has to evolve as these studies move forward. I understand that when talking about pathological predictions, a lot of it's based on clinical assessments, which I think is very fair. But I think functional laboratory assessments like what we do has an important impact, and and that there's probably a diversity of functions that should be incorporated.
This made me think that probably in 5 years or 6 years, we're going to have to kind of recheck the function of the protein of our kids, kind of individual. Do you think that's going to come? And because it's a precision medicine question, then I guess you have to be more precise at some point. Do you think that's coming on the pipe then?
Yeah. This is the future, and things are moving faster and faster. The ability to do more and more high throughput studies like this in larger volumes it's becoming faster and cheaper. So, sure.
Are you planning on doing more, putting more stop mutation, protein truncating variants through your thing? Just because the three had two had one phenotype and one had a different phenotype.
Yeah, we have some, and I think we have 10 more in hand actually that we'll probably look at.
Great.
All right.
Yeah,
So,
sorry.
No, go ahead, please.
No, things are always surprising. We never expected the diversity in the general population, and we didn't expect the stop codings to really show much. So we initially throw in a small number, and they're like, initially with our population controls for their first study, we put in 3. We thought, they're all going to be a wild type. We're just wasting our time. And they all had very diverse responses. So we're learning as we go along.
So I warned you before they got on that Hans and JR would beat you up in a good way. So I'll give you a softball. From where I sit as a non-scientist who talks to the 762 patients we've counted in the world, 215 in the US. There's a 2019 paper out of Invitae that says Syngap was their 10th highest hit with 39 pathogenics or likely pathogenics. But for those 39, they had 79 VUSs, right? So my world is one of like, you've got a pathogenic or a likely pathogenic, and then we pull you into our digital natural history study, where, by the way, we have over 100 Ps and LPs with complete medical records available for researchers when you want to tie up patient data. And we have a list of variants that JR's probably already sent you. But on top of that, I have all these VUSs who are like, what do I do? My kid sounds like a Syngapian. And kids with NDDs often, they sound similar, but when you start getting into certain things, we're starting to say, maybe you are a Syngapian. And then I watch your presentation, and then there's the missense people who have been told that everyone's haploinsufficient, but we're not sure about them. And so everyone's got their own genetic report, which starts to form their identity within this Syngap community, right? And then I watch your presentation, and I kind of want to say, well, we're actually, those are all just highly, highly educated guesses. But until we are able to run each of these mutations through multiple assays, we're not exactly sure. And I guess it's a totally unfair question, but for the non-scientific person, the takeaway from your presentation is that when I describe Syngap to people, I'm like, it's not a knife, it's a Swiss Army. It's the biggest Swiss Army knife it's one of the biggest Swiss Army knives in the brain. It does all these things, and we only know about a fraction of them. So honestly, we're still learning. But when a parent is thinking, when are there going to be ASOs or other genetic therapies available to address the lack of Syngap in the brain, and then they watch this presentation from which they take away, actually every mutation is slightly different in a number of ways. Sorry about my cuckoo clock. Where do they go from here? Is it exciting that we now have this level of specificity and we're going to know so much more about each mutation? Or is it terrifying that everything we think we know is actually just a broad simplification?
I don't know. I don't want to give false hope. As a biologist, we always expect to see complexity. That's not surprising to us. And we haven't even talked about things that SYNGAP1 has alternate splicing isoforms, and there's a lot of different things, additions. It's complicated. SYNGAP1 is not a simple protein, as you say, and a lot of proteins, and the disease associated are simplified down to a single mechanism, and a single term like haploinsufficiency is raised. And I don't know I can't, as the one doing the work and the experimenter, that's not a part of my world. We do the experiments and we see this complexity, and that complexity doesn't surprise us at all. However, that complexity is the starting point for us if we're going to try to formulate a therapeutic and decide, well, either if we want to direct a therapeutic at the molecular mechanism, if haploinsufficiency is the mechanism, I can understand just raising it. That makes sense, or if it's too much, decreasing it. But if it's something else, I would try to target whatever that is. But then putting it into the context of a circuit, you might target the downstream circuit components, or you might find the pathophysiology, and there might be a way of targeting that pathophysiology specifically, or some other higher order symptom. So there's many different ways of looking for therapy options, but it requires a very sound understanding of what's going on. So we're at the, there's a lot of known about SYNGAP, but it's a lot unknown. But yeah, it's always a long process.
Yeah, sorry, my questions aren't always fair. I'm just thinking out loud. And one more, and then JR is going to use big words again. But so the thinking that an ASO that would just upregulate both wild type and mutant alleles is likely to solve this problem, at least for the PTVs. But the more sophisticated genetic therapies that are in the pipeline that might come in and actually correct the mutation, assuming we can solve delivery, or become more appealing in the context of the complexity that your presentation so beautifully illustrates, right?
Yeah, I would say that is very attractive. And again, that then highlights the ability to find those variants that are likely to be causal from not, that are best candidates for that type of approach.
Right. Thank you. Thank you very much. This is great. JR?
I was just going to ask if for some of the protein truncating variants, if you, well, not even that, for wild type, if you would look at some of the different isoforms.
Yeah, we've thought about doing that, and we should. So that's on our list of sort of stuff to do.
It just, yeah, it'd be really interesting to see wild type in different isoforms and see how they behaved against each other in the same assays. It might give some, I don't know. It might just make it more complex, but.
Yeah, we have to think, should we take a subset of impactful variants and put them in the different isoforms, see what happens, and then move all those towards a neuronal model? Might be a good idea.
Okay, we're at the hour. Did we miss any questions?
My only other question that makes me think, when you mentioned the background, some of our kids, they will have the genetic report of SYNGAP, and then they have the buses for other, then it becomes more complex. Yeah, then, and they may have a report that is 5 years old, then it comes to the same thing. You probably have to retest it at some point, see what is true buses, what is, the background complexity. What is your thought about that?
As an experimenter, when we've thought about dealing with this, we always think about doing iPS cells as a model, taking cells from human individuals and differentiating them into neurons and using that as a basis for studying things, and that preserves the genetic backgrounds, and maybe looking at the same mutation on different genetic backgrounds. Yeah, that'd be an experimental way. The other is just a very large bioinformatic way of sort of trying to understand how, just statistically, how these different variants and different genes interact. Well, I think we're just very far off. I mean, I think this field that we call functional variomics is at, it's just in its really early stages, but in the future, we're going to want to study every amino acid, every nucleotide in every gene. And, we're trying to move through autism associated genes. And eventually maybe we'll have this huge data set to say, what's the, first we have to say, what's the impact on just that gene? And then the combinatorial impact is going to be very tough and costly and time consuming to try to do that experimentally. We're thinking about doing baby steps of doing just 2 genes, but once you start having combinations, it's huge.
Can I ask a question on just how this research came to be? I mean, P10 is a big kid, and then Syngap and I saw Safari. So are we just catching one chapter of a process by which you're going to, with Safari support, work through multiple genes? Or is this the beginning of your lab starting to become fascinated with SYNGAP1? I'm just trying to understand sort of where we are in your relationship with SYNGAP1.
Yeah, it's both those things. I mean, so we do want to go through many genes. We started with P10, and then we did SYNGAP1. Now we're doing DYRK1A as well, and then there's other genes that we're moving to. We've done COVID over the last year.
Right.
But yeah, every time we get into a gene, we fall in love with it and understand the complexities. And every protein is completely different, has its unique problems or interesting complexities. And I think once we establish a platform, we certainly are invested in developing it and using it and building on it. Of course, we're limited by money and people and time, but it's not something we just want to go march through without stopping or building.
But it is frustratingly fascinating, right? It's so complicated, and when you add in the isoforms, there's a lot of interesting questions to ask. But and when I'm talking to Hans and JR, there's like, it's endless. They keep throwing things at me I don't understand. But my donors aren't the people who support SRF aren't giving us money because they love beautiful science, although they appreciate it. They're asking the question, what is getting us closer to therapy, right? And of course, basic science informs all of this. And I guess I'm trying to understand what the future looks like a little bit, right? So in 3 to 5 years, when there are genetic therapies, will it be show us your genetic report and you have a pathogenic mutation and therefore you're eligible? Or will it be, let's look at your mutation, let's take a blood sample, let's run it through 10 assays, and there'll be some score which will tell us a variance amenability, if that's the right word, to a given genetic approach? Like, what, in your opinion, is you're in this, I think you called it something variomics, functional variomics. What does the future of giving genetic therapies to young patients look like?
Yeah, I raised this before, which is, I think if the genetic therapies are safe and effective, given that, you only want to use them on the right targets. And as we know, just because you find a variant in somebody with a disorder, it doesn't mean that that's causing it, because we at this point we know so little about what those variants do, even with a predictive algorithm that tells you something. So it'd be very important to have that pre-assessment, say, yes, that variant we think is very dysfunctional, and we think this is the molecular mechanism. Maybe you'd have a bioassay that can be performed on somebody to show you that as well. And then that would justify that as a target for genetic therapy.
Got it. Thank you. Anything else, guys?
Thank you so much.
Yeah, and thank you for tackling this huge, amazing bit of biology. It really needs someone to work hard on it, and we really appreciate that you and your whole lab are going through and thinking hard about all these things. So thank you.
Well, thanks for your questions, and thanks, Hans, for your initial question by email that really allowed me to switch my focus for my talk.
Fantastic talk.
Yeah.
This was amazing. Yeah. Very, very, very
Yeah,
nice.
I can't wait to rewatch it and really look at the graphs more. Thank you.
Thank you.
Thank you again.
Thanks, everyone.
Bye-bye.
Bye.